Emma has just immense challenges.
The reason that this whole story came about was through a very dedicated father, so Brian Broadbent. He was really the person that brought all of the researchers together on this paper.
We sort of just intuitively understood that this was a lot bigger than just Emma. She's intellectually like a 3 to 5 month old, and so she's gonna like the things that babies like. So she just loves to be held and cuddled. This sort of adds purpose to her life, and she's helping science, and we wanted to push that along.
So this is a nice illustration of how both too much of a protein as well as too little of a specific protein can lead to very profound effects in terms of neurological function. So this is liquid nitrogen. The reason we use liquid nitrogen is that it's super cold, so like Emma cells we keep in here. This is a file of DNA sequence essentially, and so this particular file is from Emma's genome, and you're looking for where Emma is different from her parents. Amazingly, only 1% of our genome makes proteins. So the other 99% is essentially the wild, wild west. So we've identified this long non-coding RNA chaser, and we know that chaser acts as a brake on CHD2. So it controls how much and how little CHD2 is made. And so one of the goals now of the lab is to design an RNA therapeutic. So this is just a small RNA or DNA molecule that can target chaser and increase or decrease the amount of chaser to then put the foot down or take your foot off, essentially, that brake, and therefore control the amount of CHD2 that's present in the cell. Epilepsy is the third most common neurological disorder, and 30% of patients who have epilepsy do not respond to current medications. And so this is really a goal of the lab to rethink about how we treat and hopefully one day cure epilepsy through gene targeting therapies.